Okay, welcome everyone to our MS Translate question and answer session. I'm Brett Drummond. I'm one of the co-founders of MS Translate, and it gives me great pleasure today to introduce our first international guest on one of our MS Translate Hangouts. This was spawned out of a collaboration when I was traveling to Utah earlier in the year, and I had the pleasure of getting to sit down with Dr. John Kriesel and Assistant Professor Kael Fischer and talk about the wonderful research that they're doing at the University of Utah, looking into viral expression in MS brain tissue. We've been featuring their work this week on MS Translate. We've had a number of videos and some paper summaries, and today we're giving people the opportunity to talk directly with John and Kale about the work that they're doing. Without further ado, I'd like to introduce Dr. John Kriesel and throw it over to him to introduce himself, and he'll pass over to Kale to talk a little bit about himself. Thanks, John.
Well, thank you, Brett. It was a pleasure to meet you when you came here to Utah. I guess you're back down under now, right?
I am. I'm back in cold Melbourne.
Anyway, it's hot summertime here in Salt Lake City, and I'm an associate professor at the University of Utah. I'm actually an infectious disease specialist. So I see patients with infections and then do some research, and I'm interested in MS from the infection sort of point of view, what infections might trigger MS. So that's where my interest has really come from.
Kale, do you want to introduce yourself?
Sure. My name's Kale Fischer, and I'm an assistant professor at the University of Utah. I met John when I was coming to the university there, and he had a very interesting set of MS samples that we wanted to look at using the technology that I'd been developing as a postdoc in San Francisco. And it was a way of looking at a sample and characterizing all the known viruses in that sample. So John had assembled a really nice set of MS samples to look at, and that's where our collaboration began. Since then, our techniques have evolved, but we're still looking for a unique viral or bacterial or just unique gene expression signatures in MS that will help us help patients. And it's exciting to talk to the MS Translate community via this Hangout, so thanks for setting it up.
Of course. So do you guys want to maybe just, we've featured the work that you've been doing this week, but do you just want to sort of summarize a little bit about how you're doing what you're doing and the preliminary data that you've received so far?
Sure. Kale, would you like to go?
I think you have a better handle on the whole project, and so would you like to start? Because I think you understand more about how the recruiting works. And
okay. Sure. Well, I'll be glad to. We have collected samples, both frozen samples from brain banks of patients who have had MS and died and donated their tissues. These are usually plaque-affected brain tissue samples. We've worked with these samples in the past, and recently, more recently, at least since 2011, when I wrote an IRB protocol and started collecting samples, we've looked at patients who've either had MS or who didn't have MS but actually developed MS and got brain biopsies. So we've worked with two sets of samples, and our work, I think, is furthest along with the previous set of samples, the frozen samples from the brain banks of tissue. We've also collected control tissue and tried to analyze our MS samples in comparison to controls so that we don't get misled. It's involved a lot of complicated, basically sequencing work. Deep sequencing or next-generation sequencing, I think, is the technique that we hope is going to help us really unravel where this disease comes from. Kale is an expert in that area, and his analytic techniques have been what we've used to try to shed light on the process. We've identified some viruses in brain, including one virus that had never been identified in brain before. That would be GBV-C or hepatitis G virus. We've also successfully identified some herpes simplex virus cases in brain tissue as well as some measles cases. So we know that our technique is capable of finding viruses in brain tissue. We have not found the the MS virus per se, and we've looked very hard for that, but I wouldn't say that we found it. What we have found is some retroviral gene expression that's pretty interesting that we've talked about recently, that I traveled to Europe and talked about recently. And we put together some work on that and some gene expression data too that I think Kale's quite proud of and I think is really interesting.
Yeah, I think something that's really exciting now is we're collecting samples from patients who are in the clinic right now, instead of just working with our old frozen samples, which came from post-mortem material. So the samples really have been different, and we have a really interesting pair of samples now where an early sample really doesn't show the signatures that we see in the later samples, and so we can really watch the progression of the disease in real active patients in the work that we're doing now. So it's very exciting.
Excellent. All right, well, we might start. I've actually got a couple of questions here that were sent in during the week as we were featuring your work, so I might start with those and then I'll throw over to Andrew and Brad to ask a couple. So the first question we got was, have you looked at the bacteria that's in the infectious theory of MS, and can the sequencing that you've already done be used to look for intracellular suspects in MS, so things like chlamydia pneumoniae, Borrelia, things like that?
Yeah, I can take that. We have seen some bacterial sequences in some of these newer samples, the ones that come from live patients, but I wouldn't say that we've really done the controls and that we're really certain that we're treating the samples in a way that will find all of the bacterial signatures that are there. So that's something we're working on right now. So more on that later. It's an active area that we're pursuing, but we had been focused on looking at sequences that were inside the human cells, and getting nucleic acids out of that tissue is really different than getting nucleic acids out of bacteria. So we want to make sure before we rule anything out that we're carefully getting all the bacterial DNA that we can out of the samples so we can examine it.
Excellent. And the second question that was asked was, does your data reveal markers or signature for leaky gut, in inverted commas, or compromised endothelial health that contributes to a weakened blood-brain barrier?
I'm not sure how you would determine that. Do you have any ideas, Kale? Would we not be looking for GI pathogens inside brain tissue if that were the case?
Well, it's an interesting idea, and I think it ties in with the first question about what we're able to say about bacterial presence in the samples, and we're not quite there yet. And as far as other molecular signatures for blood-brain barrier function, I think we can say that in the MS samples, there are a lot more lymphocytes, for instance. So that indicates some kind of difference there, but it's all at a very general level at this point.
Yeah, I think the bacterial question is actually quite interesting, particularly reviewing these samples that we've collected recently from the patients who've been in the hospital and have had brain biopsies. MS patients don't usually get brain biopsies, but if they have a really fulminant disease all of a sudden, sometimes people are worried, that the neurologists in particular are worried that there might be a cancer. So they occasionally go to biopsy, and these are the specimens we've collected. And when you look at these specimens, they really do have a lot of macrophages in them. In fact, there's far more macrophages and activated astrocytes in these samples than there are lymphocytes. There are a few lymphocytes, especially around blood vessels, but boy, the macrophages are doing something. And one of the things that they're doing is they're eating myelin. There's no question about it. So exactly why are they doing that? Why are they eating myelin? Or why are they cleaning up this mess? It could be bacterial. Now, in the past, when we examined the frozen samples, and Kale, correct me if I'm wrong, but I think we compared bacterial signatures in the MS cases to the frozen samples and the controls in the frozen samples, and there really wasn't any difference. But whether that'll hold true in these better, newer samples from these patients in the acute throes of either an attack or new onset of MS, I don't know. It'll be interesting to find out. And,
yeah, I think especially in the frozen samples, our extraction techniques were really very gentle, and we need to work on beating them up a little bit harder if we want to say anything definitive about bacteria. So in the biopsy samples, there's a lot more disruption of the tissue from the preservation and then from our dealing with removing all the preservatives. And I think that's why we're starting to see a little bit more bacterial signs of bacteria in those samples, just even at the first look. So.
All right, we might throw over to Andrew and Brad. We've got a question off Twitter as well from someone who's watching live at the moment, but we'll throw over to Andrew and Brad to ask a couple of questions, and then I'll come back to that one. Andrew, Brad, any questions you guys would like to ask for Kale and John?
Go ahead, Brad, if you would like to.
All right. Yeah. Just wondering with the sequencing. Obviously, I'm assuming is this a similar technology to what the people have been looking at with the gut biome? So they've just been able to basically do a DNA scan across all of the biome of the gut, and they've just got an incredibly large data set. Now they're working through trying to work out all the separate elements that are there.
Yeah, so in my lab we also do that type of experiment. So we have a shared resource for metagenomics that is a resource for the med school, and I also have a company that does that commercially. And usually when people are doing that kind of work, they're utilizing the fact that bacteria all share a particular gene. And they can do the experiment sort of at a 10,000-foot level, but very inexpensively by looking at that one gene, the small subunit RNA that all bacteria share. So we're also looking for, so we're doing a global analysis of the nucleic acids that we find. So we're not amplifying up just that one gene. So we're looking for all bacterial genes, assign any bacterial genes, and of course ribosomes are very common. So actually the first bacterial sequences that I found are happen to be ribosomal genes, but not from that same subunit. But when we're looking for viruses, those don't have any ribosomes, and when we're looking at the expression of the genes in the samples, again, we have to take an unbiased, broad approach that doesn't focus on just that one small subunit. But that's part of it.
I think it's also important to note that we've focused up till now on RNA. So we've tried to do RNA extractions because, there are latent DNA things and lots of DNA polluting the universe. But RNA really falls apart more quickly. So RNA, we felt, represents the metabolic activity in the sample. So if we find RNA, we think it's probably doing something or there for a reason and isn't necessarily just contaminating the sample. And also that way we were able to get around latent viruses like herpes simplex virus or varicella virus and really look for things that are either replicating or metabolically active in the sample. So it would be interesting to do more, DNA sequencing, but we haven't gotten into that just because we thought RNA was more important and these experiments are very expensive.
Oh, and if I may add, we would have never found the GB virus C in the frozen sample that we found it in if we'd been sequencing DNA, because that virus, like many viruses, never go through a DNA stage. So they have an RNA genome, and it's transcribed into RNA, and it never goes into a DNA stage. So for some pathogens, you really need to do the RNA analysis.
Andrew, do you have anything that you'd like to ask?
Yeah, thanks, Brett. John and Kale, what's going through my mind is if I think about ultimately the treatment of MS, and I simply look at it as an opportunity to prevent MS occurring in people in the future, or at the second stage, if you like, retarding or constraining MS from progressing, or finally reversing symptoms that people with MS have experienced. Do you have a vision for where this work ultimately may sit in terms of this continuum, given that it progresses as you hope it does?
Well, we have views, and I'm not sure Kale and I completely agree.
That's a good thing. It's good to have diversity of view, I think.
I will say that I think this is the technique that's going to reveal the cause of MS. I hope that we're the group that is able to do it. I think we have the best samples in the world. I don't think anybody can ever find better samples than what we're analyzing. There's a lot of investment we have, and a lot of things can go wrong. For instance, as Kale indicated, maybe we don't do the right extractions, that we miss the agent that's actually causing the disease. But when you look in these samples and you see lymphocytes streaming in, you see death of neurons, you see a wave front of disease, you see macrophages behind there, you know that the most simple, straightforward explanation is there's some kind of an infection that's causing this problem. And it's up to us to be really smart and to do the techniques and learn everything we can from whoever we need to. I mean, I literally would consort with the devil to figure out what is causing this disease. And I think we can do it, and I think this deep sequencing is the right technique, and I think we have the right samples. Now we'll just see if we're smart enough to get to the answer. That's my view.
And I think for a complementary view that it doesn't in any way, take away from John's interpretation, which is really, consistent with John's role as an infectious disease specialist. And I'd just say that we're doing the best expression analysis, gene expression analysis, that's been done thus far because of the recruiting in this study that John has set up. So it's really allowed us to look at the gene expression program of the lymphocytes that are infiltrating into the lesions. And it's a very fine-grained analysis, and no one's been able to do it in the way that we're doing it before. And I think that, infectious agent or not, or whether or not the infectious agent turns out to be controllable or critical in the later stages of the disease, it's potentially opening up pathways for investigation into their druggability and so forth that go beyond the current therapeutics that are available. So we're reaching out with this data to people that are more wedded to the autoimmune hypothesis side of the MS research community, and we're trying to get feedback from them as well. So.
Good.
Okay, so if I can just jump in as a follow-up to that sort of question, whether or not, I guess it sort of falls somewhere in between the two philosophies. Do you think using the technique that you're using and samples that you're using, that whether or not there is an infectious agent causing MS, that by the end of this study we should have a definitive answer one way or the other as to whether or not that's the case, whether we find one or whether at the end of the study you will be able to say MS is not caused by a virus?
Well, I think MS is probably caused by a number of things, and not everyone has the same cause. So that'll make it, if that's true, the answer to all of the questions that you ask is probably no.
Okay.
I think.
Sorry, go ahead, John.
Okay. Yeah, I think it's quite plausible that MS is caused by different agents. if we can't catch them in these brain biopsies of people in the acute phases of MS, I don't know how we could ever discover what's setting it off. On the other hand, maybe there is a unique gene expression pattern that Kale has alluded to. And I do think that Kale has really worked hard on the techniques, and in the last few months it's really come together. And we can see these broad expression like the one Kale showed on Monday in our group meeting. It was really very impressive. And these are the best microarrays I think that could possibly be done without actually, going in and biopsying people and doing fresh tissue. So maybe this will lead to an answer. Maybe it'll lead to drug development that is more helpful than the mouse models and so forth. But really, no matter what you do, you're still looking for a cause, something to set this off. And what could it be? Can we find it? I don't know if it'll be this grant and this project. I think the best we can hope for from this project, which will extend for another 1 to 2 years, will be to gather some candidate things for the next project, which will then allow us to focus our efforts a little more, a little better, and still do the sequencing technique, but actually develop some reagents to see, for instance, what are these oligoclonal bands reacting against? There's a number of things in the world that give you oligoclonal bands in your spinal fluid, and they're all infections. They're herpes simplex, encephalitis, they're trypanosomiasis or African sleeping sickness, they're neural Lyme disease, they're neurologic syphilis or neurosyphilis. I mean, these are the things that are known, and SSPE or measles infection, these are the things that are known to cause oligoclonal bands in the spinal fluid. MS does that. Nobody knows what the bands are against, not really.
Andrew, did you have another question that you wanted to ask?
Yeah, and something that John kind of touched on as well there. I'm intrigued. One of the things that I've had the privilege to do is to talk with Brett and his colleagues at the Melbourne Brain Centre about interaction between people affected by MS and those of you that are conducting this important research. And I'm kind of intrigued. You've mentioned as you've gone through that you started with samples from people that had passed on that had MS. You're now able to secure samples from live people with MS. And I'm kind of intrigued as to what that process involves. I have one doctor say, I think that MS patients are among the most injected people in the world, and we all kind of don't want to hear that, but I'm kind of intrigued as to how intrusive is that process and what does it mean for the individuals and importantly, what that's contributing to the outcome of the work.
Well, I think I can answer that. The reason I got interested in this is because I kept seeing patients. When I was out on the wards consulting in infectious disease, there were patients who either had MS or had some sort of brain lesions that looked like they might have an infection. So I got consulted on a number of these patients, and some of them were actually developing MS right underneath our eyes, and others had MS but had, tremendously worsening disease, a tumefactive disease in this one patient that really got my attention. After that, I decided I better write an IRB protocol and get the permission to study some of these patients. So we started with just spinal fluid. We had really no hope of securing any brain biopsies. But some of the patients did go to biopsy, and we had nothing to do with the biopsy. The biopsy was actually indicated mainly to rule out cancer in these patients, make sure they didn't have a glioma. So sometimes the neurologist didn't know what was going on. Maybe they didn't have oligoclonal bands or perfect findings for MS, and so they got referred to neurosurgery. They got a biopsy. The biopsy was done. And these are the patients that we've tried really hard to collect. But we've got, I think, 9 or 10 of them. And we're able to actually take that block that was, done, it was looked, it was sectioned, it was sent to the pathology laboratory, it was looked at there. The findings were demyelination, primary demyelination in our cases. And then we're able to take that block, chop off the oxidized top, and then get some of the rest of it with consent from these people, of course, And we don't have to go back in. We don't have to ask anybody to do a brain biopsy. There's enough of these around here that we can at least do an initial study. And we've had the MS Society try to help us find other people. We've had a couple other people from Wisconsin and Arkansas around the U.S. Tell us, okay, you can use our samples, but if we really found something interesting, I'm sure there would be lots of people all over the world. And really, we can use these samples because Kale has shown that he can get RNA out of the samples. He can extract it, clean it up, sequence it, and analyze it. So that's where we are.
That's great.
And all of the details as to how people can participate if they've had their brain biopsies done are on our YouTube channel. We posted a video with Dr. Bryce Moulton, who also works with John and Kale, giving all of the details as to how people can get involved in this study if they're interested. So have a look at that if you are interested. I'm just going to go to the question that we received on Twitter from someone who is watching, at the unaffected one. Can't join today, but would like your views on MS in those who are EBV negative like me, thoroughly tested. Obviously EBV, one of the key viruses that's been implicated in MS. Do you guys want to comment on that at all?
Sure. And then I think Kale should touch on the sequencing data that we do have on this virus, because we're not without data. What I would say is I don't know how to interpret findings of 99 or almost 100% seropositivity in patients with MS versus 95 or 97% in the general population. I don't think that's enough of a difference to account for MS. Now, it may be necessary to have EBV to develop MS, but if MS were really causing, or if EBV were really causing MS, it should be abundant in these lesions. And we've looked at that, and it isn't there. At least not in excess to the controls. Kale?
Yeah, Brett, was the question about EBV? I didn't quite hear you.
Yeah, sorry. So they said they would like to views on MS in those who are EBV negative.
Okay. Yeah. Well, it's consistent with my belief that there are a lot of different causes that are likely to set off the syndrome that we call MS. And as John was saying, the difference between the number of people who apparently have apparently had EBV based on their serology, and the people that never have had it in the two groups, those with MS and those without, there's not that big of a difference. And although it's statistically significant in some studies, that doesn't mean that it's actually biologically significant. The positive predictive value of having EBV is negligible, and the negative predictive value of having EBV is also negligible. So this very small difference in seroreactivity among the two groups is not really very helpful, I think. Almost any particulate material in the brain, whether it's a protein aggregate or a virus or a bacterial cell, if there's some of that going on, you'll see demyelination, no matter what it is. And if some of those things are seminal events in the progression of the disease process, then that would be a possible explanation for the fact that it's been difficult to identify overrepresented pathogens in these cohorts. I don't know if that's helpful or not.
Yeah.
We've been and in our data, especially in the frozen data, we've been very focused on looking for actively replicating viruses, and that's really a different question than answering the question that the serology answers, which is, has the patient ever been exposed to actively replicating DNA virus in the case of EBV? So our data are quite different than the serology data.
And far better than diagnostic archaeology, we call it serology. Mm-hmm. I think we'd all be better off in the field if we just put EBV behind us and forgot about it, because I don't think it causes MS.
Well, it does cause other diseases. So.
That's true. I've seen them.
Brad,
So
Andrew, any questions that you guys would like to?
Yeah, I was just going to ask. Obviously, I think John had explained how they've seen the macrophages and that in the brain itself, reacting or attacking the myelin area, doing their job. Have you seen, or can you look at any data between where there's a lesion and no lesion, and see what the differences is? Because obviously, the immune system is reacting, and it's munching away on myelin and whatever is attached to the myelin to trigger it, but it doesn't uniformly attack the myelin throughout the whole body. And so I guess you're probably already well down the track looking at what is in that particular area that's not in other areas that is triggering this localized, very aggressive attack.
Sure. When we set up the study, so I'll start with the frozen brain specimens first. We set up the study, we certainly had the ability to examine that issue. We could look at diseased versus undiseased specimens in the same patient, but we decided not to do that because there was sort of emerging evidence from other groups that MS was really a whole brain disease. It wasn't just limited to the area of demyelination, and it wasn't just limited to white matter, it also occurred in gray matter. So that worried us a lot, and we thought we'd be a lot better off trying to examine diseased tissue from MS brain, hopefully at the site of disease, i.e. the plaque and the white matter, and then versus normal control. So that's kind of how we set up the study in the first place. Now, in the brain biopsies that we've got, we had one patient who happened to get 2 biopsies. She had them about a month apart, and the first biopsy is the one we've sequenced so far, and it's essentially normal. The pathology is almost normal, almost completely normal. There's a little bit of lymphocyte infiltration around one blood vessel if you look way in the corner of the specimen, but everything else is normal. Myelin's normal, the supporting cells are normal, the other blood vessels are normal, everything's normal. Now, we have another biopsy in the same patient from a month later that is not normal. It's completely, looks just like MS, full of macrophages, advancing front, demyelination, lymphocytes infiltrating. So we're interested in seeing in this patient exactly what is going on. We're going to be able to compare before and after. Now, it's only one patient, but that's because we would never ask patients to get more than one brain biopsy. In fact, the fact that she got more than one brain biopsy for us is just plain lucky. We'll have to see what it shows, but that's an interesting control group, and believe me, we've thought about it. And we didn't do it that way because we think MS is maybe a whole brain disease, and just there's lots of other disease that we can't see, and we didn't want to get misled.
Okay. If I can just follow up, talking about MS as a whole brain disease, what's your thoughts on the possibilities that certainly, if not a cause, a major factor in MS developing is the theory of the restricted blood flow through the brain, where the waste products aren't coming out of the brain, or there's a build-up of something in the brain that elsewhere in the body may be just processed away harmlessly, and where the brain, with a restricted flow, is both building up waste products and also being in a slightly hypoxic state due to not as much oxygenated blood flowing through. So what's a general idea about that aspect?
Are you referring to the venule study that was done recently? And it was a radiologist whose wife had MS, and he studied her brain. I think this is a story, something like this. And she had some restriction of her venous plexus, either her either some sort of difficulty with venous drainage of the brain tissue. Is that kind of what you're referring to, or are you referring to something else maybe that I'm not familiar with, like lymphatic obstruction?
Well, I guess, yeah, it's back to that the restriction of the venous jugular veins.
Well, I think that was studied in the
blood draining out of the brain. But also, I guess it ties in with the discovery of the lymphatic connection to the brain as well.
Well, let's see, lymphatic connection. It is my understanding that CSF is basically the lymph of the brain, and that CSF is produced in the choroid plexus and is absorbed by the meninges, I think. Anyway, there's a circulation of CSF that goes on, and you produce, I don't know, so many cc's of CSF per day. The thing about MS, and CSF is a good way to see, what's going on. If there's cells in there, are they lymphocytes, are they neutrophils, is there meningitis, is there encephalitis? And then in MS and these other diseases, these other infections I alluded to, there's antibody production. So there's intrathecal antibody production. The brain is actually making antibodies, and the brain normally doesn't do that. The brain should not make much antibody, and there should be very little antibody in the spinal fluid. But in MS patients, there is antibody in the spinal fluid, and these are these oligoclonal bands that have caused, so much concern. So I don't think, there may be problems with, obviously there's problems with the blood-brain barrier, and there's problems with myelin. But I think these problems are secondary to whatever the underlying process is. As Kale said, just about anything pathologic that gets into the brain, whether it's viruses or bacteria or trypanosomes or whatever, can set off an inflammatory reaction that is generally demyelinating. And some patients are able to remyelinate. Who knows? Maybe they're able to sterilize whatever's going on. Other patients have progression and more difficulties and recurrences. So if we could get a handle on exactly what is leading to that demyelination, I think we'd be way further ahead.
Brett, I have one other question that comes to mind, if I might. I wonder if either John or Kale or both have a view about there are different forms of MS, as we all understand, and I'm wondering whether they're forming any view about whether it's more likely that there's a single virus or other trigger for each of the different types or forms of MS, or whether they're potentially diverse viruses or other triggers.
I just don't think we know enough yet. We did focus on progressive disease instead of relapsing remitting disease initially because of our hypotheses around there being an infectious agent involved. And we thought that there would be more likely to retain active replicating virus, for instance, in the case of a progressive disease. That wasn't, we didn't hit the jackpot there. We didn't find one virus in all the samples. We found some of this retroviral signature that we talk about, and we found, an RNA virus that's lymphotropic in another case. So, now we're moving on to working with live patients, and it's a little bit more sporadic in terms of the samples that we're going to be working with. We can't be quite as choosy, but I believe most of the samples that we have that we're calling MS now, we're categorizing as MS, are relapsing remitting. Is that right, John?
Yeah. They're either relapsing remitting, or some of the patients have had just a single attack. So you could also call it clinically isolated syndrome, which is kind of a milder form of MS where patients don't get repeated attacks. But genetically, I don't think you could distinguish between the different forms of MS. So if you look at the genetics of MS, which is a very complex field, and I don't pretend to know everything about it, but I will say that the most important gene, genetic driver of MS, is this HLA-DRB1501. And this is a gene, a human gene that's involved in the immune response. It's a class 2 HLA gene in humans. That means it has something to do with directing the immune response. So MS is not primarily a genetic disease, but if it were, you would think you could distinguish between these different forms genetically, and as far as I know, you can't. And I don't think we can either. When we include these subjects, we just look for people that have evidence of primary demyelination on their brain biopsies and then a compatible history. And by the way, we think sometimes the neurologists don't exactly agree with what the pathology shows. So there are forms of the disease that are tricky, and not everybody's going to have the same disease.
One thing that's nice about looking at these patients that are early in their disease process is, I think it gives us a better chance of identifying causal patterns and causal agents, because there's a chance that things that were the initial cause, the seminal cause, are cleared by later points in the disease. So.
I might just jump in the question probably for Kale, I'm guessing. So when you get these samples and these sequences, my understanding from our conversations is that then they're searched against your database of human and viral and bacterial known reads. How many of those sequences don't match anything that we currently have in those databases? Do you have any idea what that might mean?
The space DNA?
Yeah.
At least 10% usually. But it's, we don't spend a lot of time agonizing over those. If we can't identify them using our normal methods, we more or less move on. There are some more modern methods where people are categorizing those sequences broadly as likely to be viral or likely to be bacterial, but it doesn't get you very much. You have the intellectual satisfaction of making the classification, but since you don't really know anything about the virus or bacteria that it's actually coming from, there's not very much you can say. And that's not to say that those methods are right out. It's probably worth going back when someone has the time and analyzing all those sequences again. And I think most labs feel the same. Is well, we don't really have time to look at those right now, but we're going to keep those on ice and go back and look at them someday. And people that are smarter than us will hopefully come up with methods that can tease out useful information from them.
Mm-hmm. I guess if there was an unknown sequence that popped up repeatedly in MS samples and not in the controls, that would be a good motivation and go and chase that up.
Yes, that really would be. And I can't say that we've completely ruled that out.
Okay. And I just have one other one that I wanted to ask. Obviously, one of the things that you've both sort of talked about a little bit today is getting samples, and obviously that's a difficulty in any sort of research, especially when you're trying to use human samples, and obviously when you're trying to get brain biopsy samples, that's even more difficult in people with MS. Is there any potential in the future to look at getting these sequences from other forms of tissue, like blood, CSF? Is it the technique at the moment that's limiting with that, the sensitivity and the amount you can get out of it, or?
Well, people have been doing that. That's been going on for a while, and I think that one of the things that makes our research uniquely valuable is that our sequences are actually from the lesions, and so the pathology that's going on there, whether you attribute it to gene expression in the lymphocytes at the site, or whether you attribute it to pathogens or some combination of the two, the samples that we're looking at now really give us the most power in looking at what's going on in the brain. And I just say in reference to, if there are smarter people out there who want to look at our sequences, because we're funded by the United States National Institutes of Health, all of our sequencing data are publicly available after we publish. So if someone does have some time on their hands and they want to go back and solve that problem we were talking about earlier, they can download the sequences themselves and do that.
All right, maybe we'll just throw over to any final questions from Andrew and Brad. It's pushing on for almost 8 p.m. on a Friday night in Utah, so we'll let these guys knock off for their week.
Exactly. Nothing for me. Thanks, Brad and Johnny.
Last question.
No, I think we've picked their brains more than enough. Thank you very much for answering the questions thus far. It's been really interesting, and it sounds like really, really good work. So yeah, power on. It sounds great.
Yeah.
All right. Well, then we might wind up here. Just to echo the thoughts of Brad. Thank you, John, and thank you, Kale, for taking the time tonight to talk to everyone. From taking the time in February to meet up with a random Australian that got in contact and said, "Hey, if you got time to sit down and have a chat," I can safely say that both of them were extremely welcoming to me, and really took the time to sit down and talk about their work. And the work that they're doing is fantastic. It was a pleasure to be able to witness what they were doing firsthand, and we'll certainly be staying in touch and following where this research leads to, because hopefully there's some exciting developments in the future. So thanks again, guys. It's been a pleasure talking, and we'll talk soon.
Hope to see you soon.
Thanks, John and Kale and Brett for hosting as well.
Thanks, guys.
Thank you.
Bye-bye.
Bye.